Morning, everyone. Okay, I'm going to try to stand during this, but I might need to sit. Side off has aged me. Hold on a sec. Okay, so as a complement and counterpoint to Greg's talk, I will be talking about not the life of the cells, but the death of them and how to understand that with mass cytometry. Going over some stuff you guys probably already all know. Apoptotic regulation is really, really complex, and it brings in a whole suite of factors that are outside of what we, or the kind of apoptosis community, considers the canonical apoptotic cascade. You have NF-kappa B, you have junk, which I don't even know. Yeah, it's on here. You have ERK, you have PI3 kinase. You have all those sorts of things that people know are implicated in apoptosis, but don't know how exactly they fit into the apoptotic picture. Another kind of wrinkle in studying apoptosis is that it occurs asynchronously. This is Trail, which is a TNF family member that was thrown on some HeLa cells, and then with single-cell microscopy, they looked at how long the cells took to die in response to Trail. And you see that there's extraordinary variation in the amount of time this takes to happen, even though all of these cells saw roughly the same amount of Trail at the same time. And in this pre-MOM phase, this is before the mitochondria is permeabilized for those apoptosis aficionados, and this is the post-mitochondrial phase. Both show incredible variability. This is an interesting way to present this data, because they show that there's a lot more variability here than here. But if you took the longest time pre-MOMP and the longest and shortest time post-MOMP, you'd see that there was actually about pretty similar levels of variance. So throughout the process, there's a lot of it's incredibly asynchronous from beginning to end. And an issue with having apoptosis be complex and asynchronous is that that makes it very difficult to measure by traditional methods such as Western blotting, and also for more advanced methods. If you're using a microarray, the same problems generally still hold. So this is a cell line that's been treated with Trail and some Western blots taken from it, from different signaling molecules, transcription factors. And if you look at total ICAP B, the trend's pretty clear, right? It starts off really high, and then as NF-kappa B is activated in response to Trail, it starts to taper off. But I'm using this not because NF-kappa B is really important for our purposes, but just as a way to illustrate the problem. Because you look at that molecule, and you look at just a single point on this whole Western blot, and you find that if you were to blow this molecule, I redid this experiment, if you were to take the single point and then put it in a one-dimensional look with single cell data, you actually notice even there's a lot of complexity that's going on. There's a big distribution in terms of ICAP B alpha expression, and there's this part right here where it looks like actually there's none, and then there are molecules over here where actually there's as high total ICAP B as there would be in any other place in this Western blot. If you expand this out into 2 dimensions, which is still far below what we're usually doing for mass cytometry, you notice that the picture is actually really complex. There are 3 populations, and cells are actually moving between all these 3 populations as the process is occurring. So it's not really that ICAP B is just going down. It's that it's circling around this network. And I could show you some other work here, but it actually looks like it's moving from here to here to here. And none of this complexity is caught in this gel. So people have used other methods to look at apoptosis with single cells. But basically what happens is that you take this really complex, nuanced network, and you boil it down to 4 or 5 markers that you call sort of your canonical apoptosis markers, and then you just kind of assume that everything else that's going on in the process is captured in this relatively limited view. So you get a pretty small sliver of what's going on. And this is actually, if you guys are more interested in this work, this is from a really good paper from Sabrina Spencer from Harvard in 2009 in Nature, which does some of the best single-cell apoptotic work. But although that's a great paper, I would argue that new technology is really needed to understand the complexity of this regulation. You need more than 4 or 5 markers that you can look at the same time. You need to do that in a physiologically relevant system, and you need single-cell resolution. So I bet you're all really curious about what the technology is that would be able to do this. CyTOF. So this, in all seriousness, CyTOF has been a fantastic tool. In the couple years I've been working here to understand apoptosis. And I know there are some specific questions about apoptosis, and I'd love to talk to people after about kind of where they're at, what tools they've been using in order to look at apoptosis. Because even with the CyTOF, it's still a pretty complex system, I think, to try to unravel. But with use of the CyTOF, you're able to get a picture of not only apoptosis, but cell survival, survival, cell cycle, DNA damage response, proliferation, stress response, in order to get a really global picture of what's going on with this process and how it's regulated. So we have, this is a partial list of the antibodies that we have, and if there are specific questions, I'm happy to talk to people after. But we have really great antibodies against BCL2, BAX, cytochrome C, SMAC, cleaved caspase 3, cleaved caspase 7, things in the NF-kappa B pathway, AKT, JUNK, and more. So we've been able to capture everything that's going, or a much more nuanced picture of what's going on in this network than people have been able to before. And adding on to that, as you've seen in Greg's work and maybe in other talks today, we have enough bandwidth that you can look at all of these markers and get a really complex view of what's going on with the network, while also using surface markers in order to do this work in primary tissue or whole blood. So I'll just pause for a second here. I think, did you already show this slide, maybe? You guys already saw this? OK. Anyway, so cisplatin, live dead stain, selectively permeabilizes dead cells. It's good over 25-fold concentration, which makes it really handy when you're adjusting your experiments at the last minute. And this is, you can use it on primary cells, and we validated it. So cells, if you give them provanidate, will differentially signal whether they're cisplatin stained or not, suggesting that it does actually recognize live cells. And this has been a fantastic tool for understanding cell death, and a necessary one. So, OK, I just wanted to give a little plug there for the method. I hope when everyone gets their own mass cytometer, you will use cisplatin. So just to go over some quick stuff about how we're implementing these apoptotic tools, the experimental design is really straightforward. You set up a time course. You either reverse or forward time course, add drug to your cells over that time, usually get some coffee in the middle. And then right before you stain them, you just throw on your cisplatin in a protocol to be released any day now. And then you guys know this. You did this yesterday. There's just the standard sample prep, fix, perm, stain, nothing fancy. And through this process, you're able to capture an incredibly complex apoptotic network in primary tissue. So this is just kind of a first glance. These are PBMCs that have been exposed to staurosporin in this case. And then we're looking at cleaved caspase 3 expression. I guess you guys have a little spade intro, but the intensity of the color is the level of expression of the marker. So the Redder these are, the higher the amount of cleaved caspase 3. And just to take you through this in a little more detail, this is normalized 0 hour, 6 hour, 12 hour, 18 hour. And you notice flipping through this, these hematological subsets undergo apoptosis in really different ways and at really different rates. And if I have some more data that shows the complexity here, but as you see, these CD4 T cells and KT cells are cleaving their caspase 3, so undergoing the later stages of apoptosis much more quickly than these myeloid cells or B cells. And so the kind of further implication of this would be that you could use a mix not just of PBMCs, but of diseased tissue and normal tissue, and then disassociate how those diseased and normal tissues, like leukemia and whole blood, undergo apoptosis differentially, with, I guess, sort of obvious implications for drug development. And to just show you kind of the kinds of tools we're using to dig a little bit deeper into this data, you can use covariance maps in order to pull out the relationships between all of these markers in these different hematological subsets. So not only can you subdivide your data into here, but then we're able to understand how all of these proteins are related to each other throughout the apoptotic process. And within each one of these, we can make a little flip book to look at how the apoptotic network is changing in response to a stimulus or an apoptotic death stimulus. And then these covariance plots can be turned into network maps. And so you are able to get a network picture of each one of these hematological subsets undergoing apoptosis, which is a real advance over some of the other techniques that people have been using, even to understand apoptosis in cell lines, never mind in primary tissue. So OK, that's my spiel. Big thanks to the lab. This has been a great collaborative project so far. And yeah, if you have any questions, go for it. Yeah.
So Greg mentioned the robustness of some of these markers,
Mm-hmm.
the fact that maybe after a few days they just kind of disappear. So are there any markers that can last for, let's say, 3-4 days that are robust enough to last for 3-4 days or so?
I'm sorry, I don't understand. You mean post-stain?
So the idea is that some of the markers just some of the cells just disappear, the markers just disappear. Right?
I think I understand what you're saying. Well, usually for looking at apoptotic stimuli, I do these things over 4 hours, 6 hours, 12 hours.
Okay.
So over days, I don't know.
Okay.
I would imagine that if you were to actually, as you add drug, like something that you think will create apoptosis in a big subset of your cells, over something like 72 hours, you're going to kill most of your cells. So you want to really capture that first moment when stuff's happening. So in that sense, yeah, the markers will disappear, but it's really just the cells are dying in response to a stimulus. Does that answer your question? We can talk more after if you want to talk more about it. Okay.
When this is platinum, you kind of get which cells have completely died, and you do end up counting them as completely dead.
Yeah.
There's not many technologies that let you look at the biology of proteins that have been cleaved up into a million pieces like that.
Yeah, and in the later stages of apoptosis, there's kind of just like this orgy of cleavage where everything in the cell starts to go down because things just get sliced and diced because there are so many caspase substrates in the cells. So yeah, it's very hard to get sort of biologically meaningful information after cells have been exposed to some apoptotic stimulus for a long period of time. If that makes sense. Well put. Yeah.
Can you go a bit more into how you're generating the network graphs from the covariance plots?
Yeah, so basically there's a process that I didn't really go into full detail here. I've been working with a bioinformatician in order to sync the cells in silico, basically lining them with some very clear apoptotic markers, and then using that process in order to recalibrate real time into I've been using this with trail stimulus, so trail signaling time. And then with each one of those, what are called kernels, which are not really bins, they have a more complex shape, mathematical space, will then pull out those covariance plots, which are actually precision matrices, so it's the inverse of the covariance. And then from there, look at those, pull out those kind of plots. If you have more questions, we can also talk about it later. Yeah.
Follow-up question.
Sure.
At the beginning, you always start with this network that are directed graph.
Yeah.
But at the end of the day, the networks you get are just covariant, well, correlation.
Mm-hmm.
I mean, usually this will not probably be causality.
Sure. Those actually, there weren't arrows. So there wasn't a. They're not directed. The edges are just covariances between the processes. That's a good point. No, that process in itself will not give you causality. So it's a great question because what I'm in the middle of doing now is a large inhibitor study in order to knock down upstream signaling molecules and then see how those affect the apoptotic process in order to get a sense of how the causality is occurring or what's causal in the process. Yeah.
Have you guys looked at different sources of apoptosis for the same cells, like radiation versus some other forms of DNA damage versus something else?
Sure. For the sake of simplicity, I've only been doing chemical stims, and also because the idea of this is to move towards maybe some sort of drug development process or have that as a possibility. So radiation would be another interesting thing to do, but I haven't looked at that. But yeah, different apoptotic inducing chemicals introduce apoptosis in radically different ways. And that's another thing that we're interested in kind of trying to uncover. Yeah. Any other questions? Okay, great. Thank you.